Hello everyone. Am I audible? Good evening. Am I audible? Please, can you kindly confirm? All right. So is my screen visible to all of you? So how are you all doing? How's your preparation? After our first discussion on replication and DNA repair, did you go through those topics? Did you revise? Did you learn? What's your feedback? Let's see. Okay. Yeah, now is it coming clear? I hope there is no more echoes now. All right. Okay. So, as I was asking, have you gone through that chapter? Replication, DNA repair. We have discussed 10 questions, right? We had discussed it 10 questions, and many of you said that yes, it was very beneficial. So, what's your feedback? See, I hope you are also studying simultaneously as we are discussing it, right? So today also I will be discussing a topic from unit 3. Okay, this is your 3B, and I will be discussing questions from transcription and post transcriptional modification. And today also I have 10 questions for you, like how we did it in the previous session. Note preparation is tough task. Well, don't prepare the notes. Please don't prepare the notes because you're wasting your time. Okay, just jot down the major informations in points, okay, or in kind of a diagram. Don't sit and write pages and pages. Okay, that is what we all say to our students. No point in writing pages and pages. All right, continue your preparation the way you are doing, and of course do it in a very strategic manner. If you want to uh, understand that what do I mean by strategic manner, then you can go to our webinars where we have discussed strategy planning. Okay, how to study in a smart way, how which kind of tools you can use to study in a smart way. You can go and check those webinars. It is there in YouTube. Okay, we have done it. So apply those techniques for your preparation. So shall we start with this new topic today? Are we all ready to discuss these topics? For those who have already studied these topics is well and good. For those who haven't yet studied, I will be definitely discussing the points as we go along with the questions. Okay, so remember those points. Later on when you do study them, study the chapter, then keep those points in mind. Okay, so let me see. If you are ready, type BIO, bring it on. Let's see how many of you are ready. Okay. All right. So let's begin the session. And the first question on your screen is: a researcher wanted to identify the enhancer sequence of a newly discovered gene. Shown below are the relevant regions of some of the reporter constructs the researcher designed to identify the enhancer. Which of the above construct can be used to identify the enhancer? So this question, it is focusing on the concept of enhancers. What are enhancers? How do they work? What is characteristic? That everything you have to study, and also you have to use that concept to understand how your enhancer is triggering the promoter. So if it is triggering the promoter, then what kind of result will you get? Like you are using a reporter gene, right? So what is the function of the reporter gene? To show the activity of a, a gene, right? To show that yes, to report, I should say, to report that whether a gene is functional or not. So if your enhancer is working, if the enhancer is good, I mean it's active, then it will trigger the promoter sequence, right? All the activators which are binding there, that will help to trigger the transcription. Okay, so in that case you should get a positive signal. But in order to construct this entire experiment, you need to know where you have to put the reporter gene. So use all your concepts and try to figure out the answer here. This is a Parsi question, but I will say that this is quite an easy question. Okay, here let me just give you one more concept here. Though for those who don't know this SA, splice site acceptor region. Okay, this is a concept which is connected to your splicing of your mRNA. Okay, what is splicing? It is the removal of your intronic parts. So our eukaryotic chromosome or eukaryotic DNA, it consists of introns also and exons also, right? So exons are the coding regions, introns are the non-coding regions. So if you want to if you're trying to get a RNA from where you're going to get the protein, then you need to remove the intronic parts from there, isn't it? So that removal of the intronic part, that mechanism is what is known as splicing. All right. And splicing is mediated by some spliceosomes who recognizes certain sequences. So in those sequences, you have a splice site, A site, okay, which is having a OH group, and that helps to initiate the splicing. All right, this much I can tell you right now, because this entire splicing mechanism is quite long. So if I go into the details of it, obviously I will fall short of time. Okay, so that is your splice site acceptor. Okay, if I just do a rough diagram, then this is your you have the 5' splice site here, 3' splice site here, you have the A site here. Okay, this is your intronic part. From here you have your exons, here you have your exon. Okay, so this intron is going to be cleaved in the form of a lariat, and these two exons are going to get joined. Okay, so now use this concept here. Okay, I'm already getting some answers. So some of you are saying that it's option 3. Some of you are saying option 1. Come on. Again, I'm telling you. Everyone try it. Okay, I'm giving you hints. Everyone try it. Okay. So let me give you the answer. The correct answer for this is option 3. Why it is option 3? See, you have a promoter here, okay, in the first case, in the first picture. Downstream of the promoter, you have the reporter, isn't it? This is placed at the downstream part. So obviously, if this promoter is triggered by the enhancer, then transcription will start. If transcription is starting, automatically the transcription of this reporter gene will start, because transcription is occurring downstream of this promoter, right? So the reporter gene will be the first gene to get transcribed, and hence I will get a positive result from here. Okay, so I will understand that, okay, my enhancer is working. Look at the second picture. So I will get a positive result from this construct. From the second construct, there is only the reporter, there is no promoter. Now enhancer doesn't work on the reporter. Enhancers are sequences to which the activators will bind, and they will interact with the activators and the transcription factors which are sitting at the promoter. So you need the promoter site. So over here there is no promoter, upstream or downstream, nowhere there is a promoter. Okay, so this is a wrong construct. In the third case, as I told you, this SA is a splice site. Okay, so that means what? I have my one of my exon here, another exon, say it is here. Okay, starting from here, this is my splice site here, this is my another exon which is starting from here, and my promoter is somewhere here, right? So if my promoter has got triggered, then obviously after I have spliced out this intronic part, okay, after I have spliced out this intronic part and I have joined the two exons together, I will get a result, or I will get an expression of this reporter gene. Okay, so that is why both A and C is right. A reporter gene is not getting spliced out, it is there in the exonic section. Okay, so this reporter gene will be present, and at time it will show some expression. Understood? So the correct answer here is option C. Good try, everyone. At least you have those who gave me 3 as option, very good. Those who gave me 1 as option, at least you have got understood 1. Okay. Yeah, promoter is not in C, but since they have mentioned this SA, no, so it is understood that somewhere here you have the promoter. They have given you the intronic section, and you have they have given you the exonic section. So upstream exon must have got a promoter attached with it. Okay. But in this case, they haven't given anything. So whether this reporter is, what is the location of this reporter, you don't know. In this picture, the location of the reporter is controversial. Okay, that is why you can't take this into account. Okay, next question. All of the following statements about bacterial transcription termination are true except, be careful. So you have to find out what is not true. So some terminator sequences require rho protein for termination, inverted repeat, and T-rich non-template strand. I hope you remember the structure of the termination sequence. So T-rich non-template strand define intrinsic terminators. Rho dependent terminators may possess inverted repeat elements, and news A is necessary for intrinsic transcription termination. Okay, you're telling me option 3. Fine. So those who have forgotten about the structure of this termination, there were 2 terminations, right? One is a row dependent, another is a row independent termination. So you need to recall both these termination process. For row dependent, you need a root site to where the row proteins are going to come and bind. They are going to move upward, right? And row actually works in a form of helicase, so it separates the 2 strands and thus ends the termination. Okay. Now, in this row dependent termination, you can have some hairpin loops. Okay, it is not mandatory, but yes, some terminators does have hairpin loops. Okay, so this is correct. Row dependent terminators may possess inverted repeat elements which may form the hairpin loop. Okay, it's not compulsory. I'm telling you, it's not compulsory, but yes, you can have. Now, repeat and T-rich region, obviously that dependent sequence where you have, if this is your termination site, you have the T-rich region here, okay, against which you had the A-rich region here, and you have the mRNA which is bound like this, right, and it is forming a hairpin loop structure like this. And here what you have is a U. This AU sequence, this is a weak bond, right? So that is why the termination, the mRNA can easily fall off after termination. So this is also correct. And obviously the first one is correct. Some terminator sequences require row proteins for termination. Okay, so all the three statements are correct. Now coming to the fourth one. Fourth one, news A. This is a protein. Okay, now this protein, this is a protein which has been found to support the intrinsic termination. Okay, because it goes and interacts with the structures which pauses the termination, which pause and also cause the termination of the signals, and it mainly associates with the row dependent termination. But it is not necessary. It has been seen that yes, it is favoring, okay, it is supporting, but it is not necessary for intrinsic transcription termination. Understood? Now see, so the correct answer is option 4. Now you may tell me, but ma'am, we always just study about the row and we just study a row dependent and the row independent factors. We don't study about anything else, right? That is the main thing we focus on. Then how to understand whether what is the functionality of new say or something. See students, what did I do? I first tried to find out which are the correct one depending upon what I knew, right? I know the row independent, I know the row dependent termination in details, I know that. Okay, so it became very easy for me to identify the correct ones. Even if I don't know news A and the functionality of news A, still I can easily eliminate this option because I'm confident that these three options are correct. Okay, now one option has to be wrong, so obviously that will be the news A. See, these are some tricks, you have to apply in your CSIR net. Okay, it's not possible. There are endless information. Okay, because people are still researching on these topics. So you get endless information on this. But the basic information, if you know, okay, the row dependent, row independent, that is the basic information. If you know that, still you will be able to get the correct answer. Okay, that is how the CSIR has designed the paper. They are not expecting you to go through all the journal papers, all the latest publications. No, they are not at all indicating. They are not all asking you that. But they are trying to see if the basics of yours are clear or not. Okay? So that is one way you can eliminate. So always find out the odd one. Now, going to the third question. Following statements are made about double strand repair model of homologous recombination. Okay. Homologous recombination is not a part of your transcription, definitely, and or your transcriptional modification, though our main topic today is transcription and the post-transcriptional modification. Still, I have included one topic from your repair and one question from your replication because just for a revision purpose. Okay, last class we had discussed on these, so that's why I've included two questions. So one question, you have it in front of you, talking about the uh, repair. Okay, the next question will be on replication. Then again we'll get back to the main topic of today. So let's see if you have done your homework properly, if you have revised those topics which we had discussed in the last class, you should be able to answer this. This is your self check. This question is for your self check. So process of DSBR recombination is triggered by introducing a double strand break in a DNA duplex in a process known as 3' end resection, and the endonucleases along with the DNA helicase degrade one strand on either side of the break and generates 3' single stranded termini. One strand of the donor duplex is displaced due to formation of heteroduplex DNA and generates a displacement loop. Branch migration allows the point of crossing over to move in 5' to 3' direction of recipient strand, and completion of DSBR recombination may generate either crossover recombinants or non-recombinant product. You have to find out which is the combination, which combination of statements is correct. Okay, and the options I have given here. It is at this side of your screen, on the right hand side of the screen. So recall the two repair systems: non-homologous repair, homologous repair. Though over here they are only focusing on the homologous recombination. So what should be the answer? Any answers? Okay, I'm getting answers. Anchal is saying option 3. Others? Okay, the correct option is option 2. Okay, it's good you're trying. All of you are trying. Good. So correct option is option 2. Why? Because see, in double strand recombination is triggered by introducing a double strand break in the DNA duplex. So this is obviously correct, right? Double strand break. Hey, that is why we are going for the double strand break repair. So this is true. Now the next one, it was in a process known as 3' end resection. Okay, you are generating a 3' single strand termini. This is not related to your homologous recombination. This is rather related to your non-homologous end joining. Okay, this is related to your non-homologous end joining. That is why I told you, revise, recall both homologous recombination and your non-homologous end joining, because this is what resection, 3' resection, this is dealing with your non-homologous end joining. Now, one strand of the donor duplex is displaced due to the formation of heteroduplex and generates a displaced D loop. This is definitely a homologous recombination. Okay, so this is correct. Next, branch migration allows the point of crossing over. No, this is wrong. There is no branch migration that is happening. That branch migration happens in normal homologous recombination. Okay, but over here you are just moving down. Okay, you are just moving down, finding the homology, you're repairing the gap, and you're coming back and joining with your own strand. Okay. Now next, so A is correct, C is correct, and the last one, completion of DSBR recombination may generate either recombinant or non-recombinant products, which is true. Okay, you can get a recombinant or you can get a non-recombinant product depending upon what gene or which allele you have. So that is why ACE, this is correct. Next question. Question number 4. So those who are getting confused, please go back and revise your recombination. So I have again another transcription, then I will give you the replication, okay, for a revision. So getting back to the transcription question. Which one of the following statements related to transcription and processing of mRNA is incorrect? So during prokaryotic transcription, DNA binding properties of RNA polymerase are altered by sigma factor. In eukaryotic transcription, synthesis of rRNA, mRNA, and some small RNAs occurs by RNA polymerase 1, 2, and 3. Splicing observed in tRNA involves successive sequential cleavage and ligation reaction, while pre-mRNA splicing proceeds through lariat formation. mRNA with premature stop codons are degraded by nonsense-mediated decay, and mRNAs without an in-frame stop codon get accumulated and translated in the cytoplasm. Now here again, okay, first you answer, then I will say this. Try the answer. If you have the splicing concept clear, splicing of mRNA, splicing of tRNA, how tRNA is processed, how mRNA is processed. See, after you go into this depth of study, okay, you will find these questions to be quite easy. Maybe right now you're thinking कि कहाँ से आ गया ये question, you know, these are so difficult, but no. And this is one question, again, you can go by the elimination process. Like how we did for that question, the news A protein, here also you can apply that elimination process if you know the details of splicing. Okay. So the correct answer is option 4. This is incorrect, this statement. First and foremost, let me get one by one. Yes, very good. Most of you have given me the correct answer. Very good. So how did you get it? Do you know about nonsense mediated decay, or you just went by the elimination process? Comment on that also. Let's see. So in the meantime, I'll discuss this part for the others. During prokaryotic transcription, DNA binding properties of RNA polymerase is definitely altered by the sigma factor, because sigma factor, we know it is very important in placing the polymerase at the right site, okay, at the promoter site. Next, transcription synthesis, eukaryotic transcription synthesis of rRNA, mRNA, and small RNAs occurs by definitely by 3 different polymerase, right? So polymerase 1 is mainly concerned with your rRNA, polymerase 2 is concerned with your mRNA, and polymerase 3 is concerned with your snRNAs, right? So this is definitely true. In the third case, splicing observed in tRNA involves successive sequential cleavage and ligation reaction, while pre-mRNA splicing proceeds through lariat formation. So this is obviously right. Okay. In case of pre-mRNA, I even showed you at the start of this this session in the first question that you are forming a lariat when you're splicing your mRNA, when you're splicing up the introns. So definitely your pre-mRNA splicing proceeds through lariat formation, but when you have your tRNA, you don't form any lariat formation. It's just the ends are getting processed, okay, and some bases are changing, like you have that pseudouridines and all, those are getting added, okay, those are getting modified. So that is what is happening, and it happens one after the other. So if all these 3 statements are correct, then obviously you will be able to eliminate the 4th statement, isn't it? Yeah, so Shoujanyo and Surender, they have answered by the elimination process. Very good. Very good. Okay. So even if you don't know about nonsense mediated decay, you don't have to worry. You just don't have to worry. You just have to know your topic well. Right? Okay. Just for your information, this nonsense mediated decay, this is actually a surveillance mechanism. Okay. Surveillance pathway that exist in your eukaryotes, and this mainly helps in reducing the errors in your gene expression, because it goes and eliminates any premature stop codon which is present on your mRNA. Okay, if there are any premature stop codons on your mRNA, then it will eliminate these mRNAs, because otherwise, after you have translated these mRNAs, mostly the products that you get, they are mutated form. Okay, so that can cause problems. So these NDMs, they are the surveillance mechanisms proteins which are checking if the mRNA is having any premature codon or not, but it doesn't tamper the in-frame stop codon. It doesn't do anything to the in-frame stop codon. It is the in-frame stop codon is there. Okay? So there is no question of having mRNA without an in-frame stop codon. This is wrong. You are going to get the in-frame stop codon. Only the premature stop codons will be removed. Okay. Moving to the fifth question. So following statements were made with respect to transcription in eukaryotes. First statement, RNA polymerase III synthesizes mRNA in the nucleoplasm. The target promoter for RNA polymerase III is usually represented by a bipartite sequence downstream of the transcription start site. The assembly factors TF3A, TF3C assist the binding of the positioning factor TF3B. TF3B is the last factor that joins the initiation complex, and phosphorylated serine residues in the C-terminal domain of RNA polymerase II serve as a binding site for mRNA processing enzyme. So you have to find out the correct combination of the statements. So this is all connected to your mRNA transcription initiation. So just recollect that part of your topic. How your eukaryotic transcription initiation takes place. Come on, this is easy. Who can give me the answer? Who can give me the correct answer? What happened? All the transcription factors got jumbled up. Now be very careful. They are talking about transcription 3. Transcription factor 3A, 3C. That means they are talking about polymerase 3, not polymerase 2. Okay, they are talking about the transcription initiation of polymerase 3. So it's not mRNA. Polymerase 3 doesn't transcribe your mRNA, isn't it? Polymerase 3 transcribes the tRNAs and your 5S rRNA. So when you're studying transcription, do go through the initiation of polymerase 1 and 3 also along with polymerase 2. Okay, now polymerase 2, everyone knows that. Okay, the transcription factors, how they are coming, in what sequence, what are they doing, everyone knows that. But polymerase 1 and 3 is also important because the CSIR asks question from this. You don't have to go into the very details, but just the promoter sequence, which factor is coming and binding where. Okay, that much will be enough. Okay, so let's see the correct option is option 2. Very good. Anchal, Suresh, Shojanyo, Tulsi, very, very good. Option 2 is the correct answer. So first and foremost, A statement is definitely wrong because it will not be mRNA, but rather it will be the tRNA, right? Not mRNA. The target promoter for RNA polymerase 3 is always represented by a bipartite sequence downstream of the transcription start site. So if you just recollect the initiation part of your polymerase 3, you will see that you have these A sites, you have the B sites, right? Or you can have the A site and you can have the C site. So these are the bipartite sequences that you have. So B is correct. Okay. The assembly factors, transcription A's, transcription factor 3A and 3C assess the binding of the positioning factor TF3B at the precise location. No, this is not right. Sorry, this is right. This is right. Okay. 3F, 3B, this is having the TBP. So with this, the polymerase comes and binds, but before that, you have to place the TF3B, okay, at the correct site, and that is assisted by your TF3A and TF3C. Now, TF3B is the last factor that joins the initiation complex. No, this is wrong. There are many other factors which joins the initiation complex. Okay, like your TBP is there. Fine. So this is not the last factor. This word is wrong. And then you have the last statement. So phosphorylated serine residues at the C-terminal domain of RNA polymerase 2 serves the binding site for mRNA processing enzyme. So this is absolutely right. If you recollect, the tail of polymerase 2 has got a sequence: tyrosine, serine, proline, threonine, serine, proline, and serine, right? Now this fifth serine, 1, 2, 3, 4, 5, this fifth serine first gets phosphorylated by your TF2H. Okay, TF2H first phosphorylase this fifth serine, that initiates the transcription. After that, the elongation factor phosphorylase the second serine, which recruits the capping enzymes and the other splicing enzymes and all. Okay, so this serine residues, they are very, very important in not only in the initiation of your transcription but rather in the processivity as well. So if you want to if you want us to discuss the concepts in very details with you, okay, point out each and every part where the question can come from, and clear all your doubts, even the doubts which you are which is arising here from this conversation, you can definitely join us. And to join us, you can avail this Watson Impact Scholarship. This is only made for you, for all this serious students who who wishes to get the concepts very clear. So here you will not only get the classes, live classes for CSIR, NET, GATE and all, but you will also get classes for your QET, IIT JAM, GATB exams. You will get access to all the global national workshops which Biotechnica will be conducting. Internship with live projects, project hand-ons are there, because we have these IISC incubated labs, which you already must know about it. So you can go over there and do the projects by yourself. And there are many other opportunities. There are certification courses. There is direct access to our coaching experts on WhatsApp. Like from the previous session, there was one student who had a doubt from doubt. So she had sent that doubt to me, and I could help her over there. So you can get access to us, okay, and you can get the VIP passes to CRISPR conference, and of course the recommendation letters and mentoring sessions as well. So there are a lot of it you can avail. So just to remind you that the deadline is 31st July. Okay, so let's proceed with the questions. So now question number 6, and as I told you, this is a question from the revision part, okay, from what we had discussed in the last class. If you have revised, if you have studied, then you should be able to answer this question. Come on, this is very easy. We'll do this is a question from replication. Again, after this, we will shift to transcription. So observe this picture carefully. Okay, look at the picture carefully. So what should be the answer? So if you have to have a very clear knowledge about what is the direction in which the helicase is moving, okay, and what is the direction in which the Okazaki fragments are being synthesized, why are they being synthesized in that manner, all these concepts should be very clear. Okay, I'm getting a lot of answers, that's great. Yeah, so yeah, many of you are saying it's option 3. So the correct answer is option 3. Very, very good. Very good, all of you. So therefore, the first question the first statement they are saying that 1 represents the leading strand while 2, 3, 4 represents the Okazaki. So that is definite. Okay, this is a leading strand, this is a continuous strand that we get to see, and these are the fragments, and the fragments are known as the Okazaki fragments. So obviously statement A is right. Looking at statement B, among the Okazaki fragments, synthesis of 4 occurs prior to the synthesis of 3 and 2. And in the statement C, they have said the synthesis of 2 occurs prior to the synthesis of 3 and 4. Now, this is where your concept lies. This is the place where the students will get confused. What we know, your if this is a little bit of the okay, first let me just draw this. You have say 3 genes A, B, C. Okay, in the first case the C has opened. Okay, so this C will be synthesized, the first fragment. After this, what will happen? Now the segment B and C will open. Okay, so C has already been synthesized. Now B will be synthesized because your polymerase is here and it is moving this side, right? After that, I will get a fork which is comprising of A, B, and C. Now, if you see that A, B, C and B we have already synthesized. Now A has got exposed. So A will be the last one in this particular series to get formed, okay, to get synthesized. So the first is C followed by B and then C. So if you take this manner, then obviously it is option 3, which is the correct one. Okay, 4 occurs prior to the synthesis of 3 and 2. Okay, all right, moving on to question number 7. So this is again another question from your splicing, post-transcriptional modification. So they are saying that in intron in a yeast, reported gene carries a mutation in the splice site branch point. To suppress the mutation, a library of point mutants of snRNAs was introduced into the mutant strain. The suppressor is most likely to have a point mutation in what? So for this, you have to recollect the snRNAs which comes and binds at the specific site. So you have the 5 prime splice site, something is coming and binding here. You have the branch point A, something is coming and binding here, and you have 3 prime splice site, something is also happening here. Nothing is binding here, but something is happening here. So these 3 points you have to concentrate on. Let's see. This is the hint I can give you. Any answers? So the mutation is in splice site branch point, which is the splice site branch point. This is the splice site branch point. So just recall the snRNAs which is coming and binding at the splice site, because this is where you are, trying to eliminate the mutation or bypass the mutation. So the correct option is option 2. You have U1 binding here, U2 comes and binds here. Okay. After that, what happens? Can you recollect after that what happens? You have U4, U5, U6, all the 3 together comes, right? The 4 lowers U5 and 6 at this position, and then U1 and U4, it just moves out. Okay. Then 6 makes the lariat, okay, and cleaves this part. Yeah, exactly. All right, I'm not going into the very details of this part again, because that is something you have to study. Okay, so option 2. Now moving on to the 8th question. In a genetic assay, randomly generated fragments of yeast DNA were cloned into a bacterial plasmid containing gene X, essential for yeast viability on minimal media. The recombinant plasmid was used to transform a yeast strain deficient in the recombination and lacking the X gene. Transformants which survive on minimal media and forms colonies should essentially have what? Very interesting. Okay. Essential for yeast viability on minimal media. The recombinant plasmid was used to transform the yeast strain deficient in recombination and lacking X genes. So what are you doing? You have the yeast DNA, you're fragmenting it, right? And then you're incorporating it into a bacterial plasmid which already contains this X gene and a portion from this yeast chromosome you are incorporating, right? Now what you're doing, you are going for a recombination with another plasmid where there is no X gene. Okay? There's no X gene here. Now, if you want, just transformation will only happen when you are going to survive first and foremost, and you need to form colonies. So, what is essential to survive and form colonies? What part of the yeast DNA is required for survivability and forming colony? Okay, Sunshine is giving me an answer. So who can give me an answer? Just recollect this fact. Just understand, like a DNA of your yeast, okay, will have what? Some intronic sections, some exonic sections, some regulatory sections, promoter regions and all, right? So you are inserting a fragment into this plasmid. Okay. So which kind of fragment will help you to survive? Will help this plasmid to replicate and form the colonies. Okay, so most of you are saying that it is centromeric fragment. Okay, Anchal is saying 4. The correct answer of this is option 4, not the centromeric sequence. Okay, centromeric sequence, it is telling you that it is ensuring the integrity of the plasmid after transformation. Okay, but that is not causing the replication of this plasmid. Enhances for the essential gene missing in the transform strain. Well, enhancers also will be able to work only if there is a promoter, isn't it? A sequence similar to bacterial origin of replication, that no, this is not going to work. Okay, because obviously in yeast you will not get a sequence similar to bacterial origin. Okay, so it has to be the yeast autonomous replicating sequence, which is the origin site of replication. That has to be present here. All right, that if that is present, then only you will be able to replicate this plasmid. So moving to the last question. Okay, sorry, this is your question number 9. Quickly, we should answer this. So during the maturation process of some RNA molecules, formation of a 2' 5' phosphodiester bond takes place. Following statements are made about this phenomena. So in this case, you have to have a clear concept about group 1 introns, group 2 introns, how the splicing happens of group 1, group 2, group 3. Okay. So because there is a shortage of time, I'm just telling you the answer here. The answer is option 3. Okay. If you see, sorry, Uh, the spliceosome mediated removal of intronic sequence occurs through the formation of 2' 5' phosphodiester bond, which is absolutely true. Okay. Uh, then you have the removal of group 2 introns occurs through the formation of 2' 5' phosphodiester bond. Only that in this case, in the spliceosome case, you have this branch point adenine, which is having the OH group. This makes a nucleophilic attack at the 5' end. Okay. And in the case of your group 2 introns, the same thing happens. There is this CAA sequence, and this adenine has got the OH group, which makes the 5' end nucleophilic attack. Okay. Okay. Now the last question. We are almost to the end of this session. So we just have a few minutes left. The following statements are related to transcription in bacteria and eukaryotes. During concurrent promoter sequence recognition and melting, melting commences with base flipping, where two bases are flipped out into pockets of the primary sigma factor. Binding of alpha amanitin to RNA polymerase 2 permits entry of the nucleotides into RNA pol 2 active site and synthesis of RNA, but prevents translocation. RNA polymerase 1 can use upstream promoter with 3 consensus sequence as well as internal promoters having a bipartite structure. FACT is associated with RNA polymerase during transcriptional elongation and helps displace the histone octamers during transcription. Okay. So here also, because of the shortage of time, I'm telling you the answer. The answer is option 2. Okay. This is true. Statement A is true because we have what in the promoter sequence we have this TA TAAT, right? So this A and this T, no, this actually flips out, and that allows the polymerase to move forward. Okay, so this is true. Now, statement B is also true because alpha amanitin it blocks the RNA polymerase activity. Okay, it will allow the nucleotide to enter, but it will not allow the nucleotides to be synthesizing, okay, forming the RNA, because it prevents the translocation. That is how it blocks the polymerase 2. It has got a huge impact on polymerase 2 as compared to polymerase 1 and polymerase 3. Now, this is wrong. It cannot, polymerase 1 cannot use upstream promoters with 3 consecutive sequences, and also this is in the case of polymerase 3, they can use it. So this is wrong. And the last statement is also right. Okay. So again, this is a part C question. Okay, you have to go into the details. So with this, we come to the end of this session and the end of the discussion. I hope this was helpful to you. Was it? So did you get an idea as to how much you have to study? Which are the places that you need to highlight? Okay, so I'm sure that after today you have the weekend, right? So you will go and study these portions. Okay? So thank you everyone. Have a great weekend ahead. And don't forget to study, and also don't forget to relax. Okay, there's nothing to get tense about. A proper strategic planning will definitely help you to study all the topics within time. So with this, thank you and bye bye.